energy. And right now, I would invite to the stage our keynote speaker, Professor Leszek Kaczmarek.
Sustainable energy production and consumption. Okay. Not for the brain. Brain consumes too much of energy, 20% of energy. Okay, thank you very much. Thank you very much for organizer for inviting me, especially Poprawka, and all the others, and to all of you for coming and giving me opportunity to present our work. I'm going to talk about our studies, and since so before you can fall asleep or get lost or both, let me provide you with a take-home message, which I'm trying to persuade you through this talk. That mind, which is from the domain of the psychology, psychiatry, whatever, is molecular in nature. There are molecules that support the mind function, either in healthy mind, learning and memory is a very good example, or disease mind, like mental illness, neuropsychiatric disorders. And what is mind? It is very interesting. This is what I found very, very well. Anyway, this in the dictionary, that the very word mind actually comes from remembering, from memory, in old German, old English. And when we look at the definition of mind has several attributes, several faculties, but always the memory is always included in this definition. But also the others, which I will be discussing today, like belief, desire, and emotion. So to reveal the most important part is actually everything about the mind is about the plasticity, especially synaptic plasticity. It's actually amazing that already by the end of 19th century, the most famous neuroscientist of all times, Ramón y Cajal, Spaniard, suggested that the memory, for us, memory is an attribute, a readout of mind, is stored as an anatomical change in the strength of synaptic connections or connections between neurons. This was Cajal who described that brain is a vast network of neurons. There's this discussion with Golgi, whether it's a syncytium or a different connection of different cells and so on. And then something Promocji, also for Mr. Ambassador, to recall Polish achievements. One of the most famous words nowadays in life sciences, for sure in neuroscience, the plasticity, has been coined by Jerzy Konarski from the Nencki. I quote this very interesting review article by Eric Adel, Yadin Duda, and Mark Myford. Eric is probably the most famous living neuroscientist, the Nobel Prize 2000. And they recall that Konarski, in his very famous book, published in 1948, proposed this expression, plasticity. So he is attributed for coining this term. And Konarski worked in the Nencki Institute from the time since before the war, as a postdoc, even Sergeyevich Pavlov in St. Petersburg. And actually, when he died, he was the director of the Nencki. It was exactly 50 years ago, 1973. So how would we imagine the brain and its function? This is a vast network of neurons. In the human brain, we are talking like 85 billion of cells, neurons. It's maybe the same number of other kinds of cells called glia. I'm not going to talk about that. And each neuron can be connected up to thousands times with different connections with other neurons. It's an immense network. So how we can visualize the brain function and then mind activity is the pathway through which the signal goes from neurons, from this neuron to this neuron to this neuron to this, or this way around. Neurons are cells. They have so-called cell body. Inside the cell body is the nucleus, where the genetic material is contained. And there are these extensions called neurites. They act as an antenna. They collect information from other neurons. And there is usually one longer extension called axon that then collects this information and sends to other neurons. Okay, so very, very simple system. And those connections here that decide about everything are called synapses. In general, synapses either can be excited, so they're called excitatory, or inhibited, so they're called inhibited. Let's focus on excitatory. Excitatory synapses are those on which very simple amino acid, glutamate, operate. It's released from so-called presynaptic site, this one that comes from axon, and then there's synaptic cleft, 20, 30 nanometers, and then there's a membrane of the next neuron. And on this membrane, there is an extension called dendritic spine, because it's like spine in rows. And these dendritic spines harbor these excitatory synapses, and they have receptors for this glutamate. Those are proteins that extend through this membrane, okay? And when they are activated, so when glutamate binds to those proteins, they open a channel, and through this channel, ions flow. Potassium, sodium, calcium, and this is what is there. Through these ion channels opening, the ions are sort of going one way or another way, depends on the concentration, and then it may excite the neurons to electrical activity. This is what we know classical neuroscience. And those spines come in different flavors and shapes. They may be small, they may be big. If they are big, called mushrooms, because they have a big kind of membrane, they accommodate many receptors for glutamate this channel. So they are easier to excite. If they are small, they are stupid. They have very little receptor, very few, so they cannot be excited. Okay? So the issue is the signal flows through the system depending on how efficient, what is the buildup of those synapses, the postsynaptic zones in particular. Yes? Are they small, signal doesn't go, or big one, mushrooms, signal goes and excites the next neuron. And this is absolutely amazing what Cajal in a way predicted. We had no idea about these things, these receptors and so on. Our contribution to the field long time ago, over 30 years ago, was actually to discover that it's not only that glutamate activates synapses, but it also activates, produces a signal, a chemical signal within a cell that goes all the way to the cell nucleus, and then can modulate gene expression. Why is so important? Biology, every cell in our body has more or less the same genetic material, but the cells differ. They differ structurally, amazingly, like neurons or liver or the lymphocytes or whatever. But they also differ functionally. They respond to coronavirus or they do not respond to coronavirus. And this is because the same genes are, as we say, are expressed, technically transcribed, in a different level, different genes at different level. So this combination of the level of expression makes all the cell bodies, makes us. This is the major thing in biology, in life sciences, okay? So showing that glutamate, the organizer of synaptic plasticity, the one that controls that synapses are bigger, smaller, and so on, produces changes in the gene expression, open the field that didn't exist until then. This is called molecular neurobiology, showing that indeed there are changes in the brain, also in the context of learning. Because we showed that this gene expression, and the gene was called CFOS, it encoded protein called CFOS, are activated during experience of learning or other forms of plasticity. CFOS is a protein that associates with another protein, so it's called dimer, called AP1, and they regulate other genes. So this was the most amazing. We discovered that during learning, there are regulators of genes that are activated. It means that the whole gene program, conductors of the orchestra, could be in place, that could be active. This opened a way to look for such genes. So this was originally what we found. That took us a long time. Okay.
No energy.
Yeah, we need to sustain it. Then we're looking what genes are activated by this transcription factor. And interestingly, we provided evidence for two of them that are functionally associated. One is called TIM1, tissue inhibitor of matrix metalloproteinase. The other is called MMP9, matrix metalloproteinase 9. What they are? Those guys, the MMP9, I'll show an example of MMP9. It's a enzyme, it's a protease. So it's enzyme that cleaves, this is why it's in the title, that cleaves to proteins. It does not destroy them really. It splits them into parts, or taking away one part, make it more active or less active or whatever. Interestingly, this is so it's produced in the cell, the process called translation, when it's hit of so-called ribosomes, is packed in some vesicles, and through vesicles is released outside the cells because this enzyme operates extracellularly. And we have provided very strong evidence that it operates at the postsynaptic site, is released from postsynaptic site, and regulates this transition from the small, stupid spines, the big one, the mushroom one, the wise one. And then, so it's expressed, is released, and then is released in so-called proform. There's a peptide covering enzymatic active site. Enzymatic active sites have to be exposed in order to enzyme to exhibit its activity. So the other proteases that cut it away, and this is active. Cuts through proteins that are around. It's a big molecule, so it cannot really go too far. We are talking, I don't know, hundreds of nanometers, no more. And then is very quickly, after a few minutes, we showed, blocked by the endogenous inhibitor of its activity, and this is the STIM1. So interestingly, both guys were regulated by our CFOS. And I already said that this is especially important, the transition from small spines to big spines. So it's a plasticity. We discovered some substrates. Enzyme cleaves through a protein, and this protein that is cleaved by this enzyme is called the substrate for this enzyme. And we found some of the substrates to be located at the synapse. Though most of the studies on MMP9 in the brain were related to the immune system activation, inflammation, and the blood-brain barrier breakdown. You know that the brain is somewhat protected, not every molecule from the bloodstream can go to the brain because there are this sort of barrier, endothelial. But MMP9 is classically involved in this inflammatory reaction. But we have some followers. We started this field of the MMP9 plasticity over 20 years ago. And last year, I've been on 2 conferences, one American Brain Research Conference, one especially one in schizophrenia. The sessions were exclusively on the single proteins. Yes, it's going on. Okay, now I would like to put the move. Yeah. So how we can study learning and memory? Very good thing is called IntelliCage, and doesn't work. No, unfortunately, it doesn't work. Okay, so I'll explain you. So there's a cage in which we have like 12 mice living happily together. They have food here. They have kind of a shelter there. We don't see what. Maybe we should actually switch the light with more dark here and better light than it's possible. And they have corners in their bottles so they can drink some. There's a better. Okay. And what we do is that the animals suddenly we change the water in one corner into sweet water, with sucrose or saccharin, whatever. This is. I say it doesn't work. Okay. So then the animals want to go more preferably to this corner. Remembers the sweetwater corner. Mice have a sweet tooth, they like sweet. The percentage, how often they go to the sweet, we say reinforced in behavioral language corner, means that they remember better or less. Normally it's 25%, more or less, they have some preference. We usually start with a corner that is least preferred, 20%, 18%, and suddenly 7%. And then we can do a nasty thing also. We can put an air pump, or we can put a water that will be bitter, like quinine. The one that is tolerant, unless with gin. Okay. Okay. So during this learning, we looked for the CFOS expression. CFOS is a nuclear protein, so it's in the round structure in the cell body. So this is when you can stain it. Same for it. This is just black dot. Okay? More black dot, more CFOS. And we found that when the animals are learning the good thing, the sweet water in the corner, there's plenty of CFOS in different parts of the brain. This is every dot is 1 neuron. but in the part of the brain called central nucleus of the amygdala, which is emotional, motivational part of the brain. Both in mice and humans. Suppose it's only when we say to preference level, the sweet water. But when we put the air path in front of the nose of a mouse, then there is no expression there. This is important for us on my talk, that I'm saying that there's some association of a very small part of the brain, called central nucleus of the amygdala, with appetitive learning, the sweet water. And okay, so CFOS is there. What about MMP9, which is regulated by CFOS? Okay. So to see whether MMP9 is important in this kind of learning, we use mice, normal mice, we call white type. Okay, they are not white mice, they are all laboratory. White type, genetically, they have all genes, okay? And we have a mutant mouse, which is a single gene mutated. We call it knockout. This is the gene of MMP9. By the way, I got this mice from a very nice lady from San Francisco, Zena Webb. And when I first met her, she said, "Jak się masz?" And this is as much as she knew. Anyway, so what happens is that the mice, in this case, were learning either that in the water, the beverage is one in one corner, the other three were empty, but one bottle was sweet water, another bottle was tap water, and the normal mice, white type, prefer to go almost 100% to the sweet water. Those missing, minus, both copy of gene are missing, of MMP9, were almost stupid, 50% chance. Okay? But if we put quinine, perfectly okay. Both of them avoided. Both genotypes, white types and so on. So MMP9 is important to this. And then coming to the central nucleus amygdala, we did a special trick. We produced nanoparticles that are slowly dissolving over days in the brain, that were loaded with a very specific protein called TIMP1, the inhibitor, the blocker. And we injected this only selectively into central nucleus of amygdala, and TIMP1 could operate only extracellularly for the next few days. And what happened is, when we put these nanoparticles in bovine serum albumin, another protein with no function, this was, again, they learned sweet water and so on. Okay, they learned sweet water, but when they were blocked with TIMP1, they did not learn. It was exactly as the knockout, really. And then, when we did this aversive learning, it means quinine, bitter solution, they were okay ones. So I think it's amazing. It's very small part of the brain, a single gene, and controls entire appetitive behavior. You can disrupt your appetitive behavior if you mess up with something over there. And this was healthy mind, learning and memory. Now disease mind. Disease mind is like mental illness. Yes. One example: Fragile X syndrome. That's a disease. It's an autistic syndrome, which is monogenic to some genetic problem. Humans, boys, especially because it's Fragile X. So boys. The activity of the gene is gone. The protein. The activity means the protein. So the protein is not animal produced. Okay? So this is like a knockout in mice. So in mice, people reproduce this, just removing this gene. That's okay. Fragile X of MRP. Okay, this is the importance, most common cause of hereditary intellectual disability in boys. They are autistic spectrum symptoms. The epilepsy fairly often. We study also epilepsy. I'm not very good at. Interestingly, these protrusions, these symbiotic spines are longer and thin. Okay, and we have previously shown that MMP9 is involved in this. So actually, Irina et al., you can guess from name, she's Ukrainian. She works in Irvine. Irina laboratory showed that when you culture these neurons from the Fragile X mice knockouts, okay, that are reminiscent of the human conditions, the spines that are thinner and longer, they actually can be normalized to the regular shape, right shape, proper shape, with minocycline. Minocycline is an antibiotic, which is tetracycline derivative, which is used in humans for acne, for example. And this asks about the role of MMP9 in the Fragile X. And we've been asked to collaborate, also for expertise with this, with another group from Sacramento, who were clinicians, and they give the minocycline to the boys with disease, and found that actually it stabilizes kind of cures them, okay, to some extent. And our part was to show what happens with MMP9, and indeed MMP9 goes down when it's minocycline present. When the minocycline is removed after like 3 months, it goes up again. Okay, there are 2 different human individuals. There are some numbers on so on. This is still continuing. Minocycline is still under clinical trials in the US for treatment of autistic syndrome, not only Fragile X, also in combination with other drugs. Neurons are the most clever cells in the body. Okay, I told you that important is the proteins. Proteins make up the cell. They function, dysfunction, whatever. Neurons have these extensions, these spines, synapses, thousands. Okay? It wouldn't be very wise to produce protein everywhere. It would be nice to have a control of a protein on a single synapse. Okay? But the genes is in the nucleus, synapse is micrometers away, 10, 20, whatever, even more, sometimes 1 meter. So what you can do, you can deliver an intermediate from the nucleus to the synapse, which is called mRNA. Yes. Okay, we should learn more about mRNA because of the vaccines. And this mRNA in neurons can travel to synapses and sit there. Interestingly, FMRP, our protein from the disease, actually this is the one that takes on the shoulder the mRNAs and delivers to the distal locations. Okay, cargo. And then it sits not being translated, that means not proteins not being produced. And FMRP blocks this local translation. This is what we show. When FMRP is missing, you will see. So this is this is MMP9 mRNA colocalized with FMRP protein. These granules carry the mRNA. And then when the mice miss this FMRP protein, there is more production of MMP9 because when this is delivered to this local distant location, is blocked. Translation doesn't occur. If MMP disappears, translation goes on and on and on. Okay. Okay, so this was a story that if you have missing MMP9 in the central nucleus of the amygdala, you have deficit in appetitive learning. Yes. Okay. What about if you may have too much of MMP9? And this is the case of Fragile X. You have too much of MMP9 because the protein is missing and so on. So we have just recently published with Ewelina Knapska group analysis of MMP9 in the central nucleus of the amygdala in mice missing Fragile X mental retardation protein, and found that indeed there is some increase of this MMP9 mRNA. And those animals are also deficient in this appetitive level. They have too much, the same. The effect is the same. Is disruption of appetitive level. This shows, this is very important lesson actually, that because people ask, okay, so pharmacologically and so we can do. With biology in general, it's fine-tuned. Okay? Having few percent less of something, few percent more of something at this special location, this special time, makes all the difference. And the effect may have the same. You just cannot mess up with those things. So when this gets stabilized with, again, the same nanoparticles with one, they were learning normal. These 2 bottles. Interestingly, Ewelina and Ala, when she was pushed, and now she's a postdoc, aspiring to have her own laboratory, but when she was a student, she developed a special system to assess social behavior. But Ewelina is most interested in social behavior. So they created a system. The problem with behavior of mice in general is that the tests are very often, but then very artificial. So this is why we go to IntelliCage, developed by a French hospital in Zurich, but then Ewelina got this one to make it more ecologically relevant. So in this case, it's again a group of mice. There are 4 chambers. They live together. There are special sensors that can measure what the mice, where the mice, each mice is there, and so on. And now is a test in which, for example, in one of the chambers, you change in a corner a bedding, from neutral bedding to a bedding that was used by other mice. So it's a social stimulus. Okay? Oh, the colic was there. Let's see what happens. Or you can measure whether mice follow the other mice, whether they want to do it together or in isolation. If they want to do it together, they are more social. And what happens is the fragile X mice, they are socially deficient. They have problems with this recognizing the bedding, and they also, which is showed here actually, and they also have problems with going one after another. The TIMP one doesn't, in central amygdala, and central amygdala is involved, Severina was showing that, but in this way it doesn't cure at all. Okay, so Ala coined this kind of idea that now we have to think about treating the symptom, like in autism, generally, specifically. We do not expect to be able to treat the entire spectrum of symptoms. Probably won't happen. We have to address specific symptoms in a specific way. Okay, alcohol. When Kasia advanced, Kasia is now professor, but was in my lab, she developed a system to study alcohol addiction in mice. So there are, again, this IntelliCage. In one corner, there is bottling. First, 2% alcohol, 4%, 6, 8, and then you drink. Yes, it's like a drink. Not all of them. The high drinkers, low drinkers, like them. But anyway, we can study the interest in this alcohol by testing the motivation by following way. So in order to get to these bottles, the mouse has to, which you didn't see, the mouse has to make a no smoke to penetrate into the corner. Okay? Now we are lost. It goes to the corner, it makes 2 notes, 4, 8, 16, 32. There is an alcohol behind the game. So how much they are eager to move, do the no smoke is a measure of motivation. 2 times, 4 times, 60, 32, 64. Okay, this can be measured. The other way, they are used to have alcohol in one corner, and now we take away this alcohol. Do they still go to this corner? Do they remember their motivation to go to the corner that used to be reinforced with alcohol? And using this system, we found that mice, those missing MMP9, okay, they're actually deficient in both tests. They are less eager to make those no smokes and less eager to search for a corner that used to be reinforced with alcohol. Okay? And then we come to our favorite central amygdala, and now we have genetic tree to substitute the missing MMP9. So-called, this again, viruses are coming here, there are so-called viral vectors that we can inject specifically the gene for this MMP9 selectively into one location. In this case, it was central nucleus of the amygdala, and bingo, they are cured. Now, if they have reinstated MMP9, they show the same level of motivation to seek alcohol as the wild types. And what is interesting, this is collaboration with Jurek Samochowicz, who is from the Pomeranian Medical University. Jurek is a psychiatrist. And okay, it's difficult to make experiments on humans, yes? So, but nature has done deep experiments on humans. One of the experiments is so-called gene polymorphism. If there is some big change, like this FMRP missing gene, we say mutation, right? But we have genes in different variants, slightly different, still more or less okay. And some of the genes may be different in that sense that the level of the produced protein may be higher or lower. So with gene polymorphism, it does not affect the protein itself, but how much we produce in our body. And there's a polymorphism, whatever that is called in -1562, far away the region that is being expressed, right? That is the higher production of MP9 is like 1/4. Yeah? And 3/4 have lower. And then they ask, they study alcoholic families. Yeah? And they look in the questionnaire how much people were claiming to be seeking for alcohol, showing motivation for alcohol, despite physical and psychological harm. Physical, hangover, psychological, husband or wife, okay? And what happens is that people who are more prone, more motivation for alcohol, have higher quality level of it. But it also concerns humans. And schizophrenia is the last example of the disease mind. It's a kind of the most emblematic mental disorder. It's a kind of a triad of symptoms, cognitive, negative, and positive, and also is characterized by delusion, hallucination, or whatever. This is something you can read. I like it, this definition. What are really delusions? Because you meet delusional people also. Despite all the evidence, they claim that something happened. Anyway, so I have a friend in Göttingen, Max Planck Institute, Hannelore Eschenreich, and she gathered a large group, 1,200 of schizophrenic patients, and perfectly matched sex, gender, education, age, whatever, group of controls. And she claims to have 3,000 data points per person. Questionnaires, DNA, brain scans, motor action, whatever. So I said, this was actually European Grand Prix. I asked Hannelore, why you do sequence for me MMP9? That's not the sequence, the gene sequence, in all those people. Okay? And she said, wow, there is a polymorphism, okay, that predisposes to delusions. In one variant, the schizophrenics were less delusional, in another more delusional, and were highly statistically significant. We never heard about this polymorphism before, okay? So we started to study. Kasia Wojpeta, who was PhD student in the lab, she looked into this, was PhD project, and the polymorphism was located. Normally, the mRNA, this intermediate from gene to protein, is organized, like in biology, everything is organized. The front is called 5', the end is called 3'. And this part that is behind the coding sequence. Coding is the one that is on the ribosomes gives protein. But there is a part at the very end called 3' untranslated region, does not get translated, that is responsible for this translocation and regulation and so on. So Kasia looked what happens. We assume that this may be involved in FMRP binding as well as the local translation of the cytoplasm. And this is what she did. She got the human gene, put into rat neurons in culture, in vitro, and looked for how much MMP9 is produced. And depends on the variant, the production was different. And then the composition of the dendritic spines between the sphelopodial versus mushroom was also different. And she did with colleagues from Poznan, by organic chemistry, the bioinformatic analysis, and they again showed that the structure is so much changed that it influences FMRP binding. And then you may wonder whether you can study schizophrenia in mice. Not easy. This is a problem we face. In neuropsychiatry, it's very difficult to discuss with mice. Yes? Normally with these diseases, schizophrenia, you discuss with patients and you learn something. It's mostly through this interview, the diagnosis. There are no biological ones. This is one of the major reasons. Okay. But one of those receptors for glutamate is called NMDA receptors. It has so-called antagonists that block and agonists that stimulate. And it was found that, so since this is a very important receptor, people studied this antagonist, the substances that block to affect some disorders. And they discovered in humans that if they are treated with this NMDA receptor for glutamate antagonist, the people get hallucinating the delusion. Okay? So this came from humans. So now what we do with this, a very simple test, we take a mouse, there's an arena, there's a mouse, we put the mouse there, and the mouse wants to explore. Wants to know what it is. Then we dig, and then so. Then we inject the antagonist of the NMDA receptor, okay? Ketamine, by the way, it's one of those substances, PCP. And then they go around much faster. It's called increased locomotor activity. Okay? What we found previously, what Kasia found, that actually this polymorphism is predisposed to less MMP9 being produced at the time, produced delusion. So we took mice, this time not entirely missing the MMP9 genes, but the wild type was crossed to knockout, and then we have heterozygotes. One gene is there, one is missing, so they produce twice less of it. Okay? So we thought this might be kind of a modeling here. And then you see those mice called heterozygotes, they actually much higher local activity. So maybe they are delusional. Okay. And okay, so now I want to say, for those who start research, my personal history of research is that I'm studying the same thing since I came back from the postdoc in the US, 86, I started to work in the Nencki. As a postdoc, I work on the oncogenes, proto-oncogenes, cell proliferation. Then I submitted publication when I got back to Poland and started neuroscience. I haven't done any neuroscience before, with the idea that might be gene expression related to a healthy mind, with the learning and memory as a reason. Okay? And ever since, well, 37 years, I'm doing the same thing. I'm going a very meandric way. It's not a straight river, but I'm coming back and forth to the same issue. So whatever we've been studying is we try to understand what is the meaning of this increased gene expression. And what we found that actually first it occurs, then it is critical. It's not that there's a transcription regulator, that it codes for some genes that code proteins, that codes for synaptic plasticity, etc., etc. So these are the major results. And we ended up with the 79. It's a very interesting molecule involved in this. I wanted to show a general story about the mind that might indeed be subserved by the molecule, that might be excellent or not excellent. I have not talked about something much more practical, what we are doing. Epilepsy, development of it, is also supported by aberrant plasticity. And MMP9 we have implicated in this operation. And we have, for example, a project with a startup company in Poland called Biuro, who got money from NCBR for 20 million to go to clinical trials with a blocker, with an inhibitor of MMP9. And this is ongoing, seems to be a very interesting after traumatic brain injury and stroke in humans. We'll see what happens. Okay, so finally, I would like to thank, this is the recent picture of the colleagues in the lab. There are many people involved in this work, both in our lab and the collaborating laboratory. Thank you very much for your attention.